Alright everybody, and welcome back to Lab 3. Today we're going to be going over bacterial identification as well as Koch's postulates, step 3. Before we get into it though, I do have some announcements to make. Regarding to the assignments that we've been giving you guys out, make sure you do both compare and contrast when we ask you to. Be very specific in the details when you fill out, when you answer. And just in case for some people that didn't know, you can really only see viruses under an electron microscope. Also, make sure you answer the questions with your own words and not directly copying the notebook, because it's considered plagiarism. And do read the questions when you answer as well. People do miss points for not completely answering what's being asked. Also know the difference between the different types of lenses that we use. Make sure you specify which lens you're referring to when you write down the answer to a homework question. I think that's it. We can just get straight into it. So today we're going to be doing three things. We're going to be doing gram staining. We're going to be doing step 3 of Koch's postulates, and we're also going to be streaking onto McConkey agar. So just before we get into it, when it comes to identifying which bacteria we're dealing with, simply looking at the macroscopic qualities of it is not sufficient to identify with certainty which genus and species of bacteria we're dealing with. The way we normally identify which bacteria we're working with is by looking at its metabolic properties, meaning what properties does it have to metabolize certain compounds. Here in the picture, we have a serological assay that's normally used, in which you take a bacteria that you're trying to identify and you dip it into each one of these little wells that contain different compounds. And then depending on the metabolic qualities of the bacteria, they will change the color of the substance that's inside of these wells, such that based on the changes that you get in color, you'll be able to determine which bacteria you're working with. So for example, here you have some NO3 that's white with kind of bluish clouds, whereas over here with NO3, you can't really see if that's NO3, you have this nice red, almost orange color. So there's a difference between bacteria and their metabolic properties. However, we're not going to be doing that today. If you take a microbiome class, you probably will. Instead, we're working with differential media, which can serve a similar role as this. And we're also going to be doing some differential staining to be able to tell the physical differences between the bacteria, rather than the metabolic properties. And just for your information, it can take 14 to 19 growth characteristics to identify a pathogen on average. So differential media, as the name implies, is a way to differentiate between different types of bacteria. Usually it's by changing since each bacteria has different biochemical properties, it's able to break certain things down, like sugars, whereas some bacteria cannot. And breaking down those sugars can change the color of the media, and based on that color difference, we can differentiate between the two different types. So today we're going to be working with McConkey's agar. It's a differential media, and it's also selective. So differential means you're able to distinguish between two different types of bacteria. Selective means you will only grow a certain type of bacteria. So with McConkey's agar, you have a lot of crystal violet and also bile salts in there. You don't need to know exactly what those are, but all you need to know about those two compounds is that they inhibit the growth of gram-positive bacteria, meaning you will not get any gram-positive bacteria growing on the plate. That's where the selective property of McConkey comes in. You can also see that there's also some color indicators inside the media that are able to be broken down by certain types of bacteria, but others not. And when it breaks down, you can see these different types of, these different colors in the colonies, and that's how you're able to differentiate between the two. So that's the differential part of the media. Any questions about that? All right. Gram staining. So gram staining is a method of differentiating between bacteria using a stain. So let's see, I don't know if you'll need to know the history of it, but it was developed by Hans Christian Gram in 1844. And it's a very fundamental characteristic of bacteria. I mean, anytime you talk to any microbiologist, everyone knows what gram staining is, because it's typically one of the first tests that's performed in bacteria, when identifying bacteria. With gram staining, we will be staining the bacteria themselves so that when we look at them under the microscope, we'll see the gram-positive bacteria will look different than the gram-negative bacteria. And you'll be able to tell which one's which based off of whether it's positive or purple or negative pink. So what gram staining does is it targets the bacteria's cell wall. The difference between gram-positive bacteria and gram-negative bacteria is their composition of the cell wall, meaning what it's made of, or what percentages of what it has, and I'll get into that in just a second. The main difference, or rather what we're focused on when doing gram staining, is looking at this layer of what's called peptidoglycan, which is a type of, peptido, meaning peptide, so it's got amino acids in it, and glycan, or sugars. So it's sugars mixed with amino acids to make this very thick protective substance. And the difference between gram-negative and gram-positive bacteria is that even though both of them have a peptidoglycan layer in their cell wall, the gram-positive bacteria has a much thicker peptidoglycan layer, and that's how we're able to tell apart the two. So I don't like this diagram very much, so I added my own that I found off the internet. Here we have the cell wall of the gram-positive bacteria. So down here would be like the inside of the cell. Up here would be the outside of the cell. And you've got the cytoplasmic membrane, right, the lipid bilayer. And on top of it, you've got this thick peptidoglycan layer. It's very large, and it's got this teichoic acid and different types of acid running through it that anchor it down to the cytoplasmic membrane. Whereas with gram-negative bacteria, you have more layers overall than the gram-positive. However, the peptidoglycan layer, which is what we target with the gram stain, is much, much thinner. So the crystal violets, the crystals that we're going to be working with, will attach to the peptidoglycan layer. And also in gram-negative bacteria, but it's going to be, you're going to have such few crystals in the gram-negative bacterial cell wall that you won't be able to see it under a microscope. And that's how we're able to tell between the two. Well, actually, another way we're able to tell between the two is we're going to be using a counter stain, which is specific for gram-negative bacteria. So we're going to be using two stains today. That second stain is called saffron. Alright, so I'm going to go into the procedure really fast. I won't go over it too much in detail because I hope you guys all read the protocols. What I recommend you guys do with really any lab course when you have some protocol to do is that you write down a list of steps of what you need to do rather than having to look through the notebook, because even though the notebook is helpful, it gives you a lot of information, it doesn't quite list the things that you need to do out step by step. So that's why I recommend you have one of these for any of your lab classes, so you can just sit right down and immediately do what you need to do. But in case you guys did not do that, I will have to go through the steps in order. So let's see. What we're going to be looking at are the bacteria that you guys isolated, hopefully in pure culture from last week. So if you remember, the first step we did was we took a swab from the infected potato slices, we grew it on media, and then we tried to get a pure culture out of them. So hopefully if you guys streaked well, then you should have pure cultures and you should have separate colonies. And that's what we're going to be testing today. You will take your inoculating loop and you will, well, first of all, you're going to have these slides over here. should have at each of your tables these glass slides that you use for your microscope. You're going to put 1 drop of water in it. Try to make the drop of water as small as possible. And then you're going to take your inoculating loop, make sure it's sterile, touch one of the colonies that are growing on your plates, and you're going to smear it into that little bit of water that you have. Now, what I did is I also took my Sharpie and I drew a circle on the bottom of it, so that way I know where the bacteria is, because after the water dries up, it's kind of hard to see it. And that should take you all the way to step 4. So you're going to let it air dry. Once it's air dried so that there's little to no water on it, you're going to pass it through the flames of your burner. Make sure you have a a clothespin hanging onto it so that you're not touching the glass directly, otherwise you're going to burn yourself. And just pass it through the flame. You don't need to hold it over, because if you do, you're going to cook the bacteria and your gram stain's not going to work, obviously. So just pass it through the flame twice, wait 30 seconds, pass it through the flame again. And we got a clock in the corner, which you guys are going to be using a lot today. So putting it through the flame is going to cause the cell wall to kind of melt a little bit, part of the components, which are going to make it stick to the glass slide. And that's important because we're going to be washing the glass slide a lot. Make sure, again, step 5, make sure you don't hold it in the flames too long either, because you can also break the glass. Make sure you guys wear goggles as well, because you don't want to get any of this stuff into your eyes, and that your lab coats are also buttoned up. If you're wearing glasses, you don't need them. You don't need goggles, but I do recommend them. So once you're done with all of that, again, you're going to have your slide on the clothespin. You're going to put 5 drops of crystal violet onto the center of the circle, and you're going to let it sit for 60 seconds. Make sure you use that clock over there. Then you're going to pour off the stain and rinse it with water. So just tilt it into these little bowls that we provided, and just make sure the stain comes off. And using the little water bottle, you're going to drop it not directly onto the circle, but rather above it so that the water slides down. And you don't want to wash it too much, but once you see that it's kind of clear and that the water coming off doesn't have very much purple left, then that's fine. And once you do that, you are going to, I would recommend you get some lens paper and then blot it dry, just so that you can get all that excess off. You're then going to do the same thing that you did with crystal violet, this time using the iodine, and you're only going to wait for 30 seconds. So those crystal violets, they're what turned they're the purple that we see under the microscope. The problem is those crystal violets, those violet crystals, they don't stick around too long, the chemicals. So we put iodine in it so that it kind of glues it together to the peptidoglycan in the gram-positive bacteria cell wall. And then you do the same thing, you rinse it off with water. And after you rinse it off with water, you're going to rinse it off with the decolorizer. That's just ethanol and acetone. And that's going to wash away any of the excess crystals that are not bound to the peptidoglycan. So this is, if you have any gram-negative bacteria in your slide, then they shouldn't have any of those crystal violets. Then after that, again, rinse it off with water. So you rinse off the decolorizer with water, blot it dry, and then you're going to put saffron in, this time for only 20 seconds. And then you rinse it off with water and allow it to air dry. So it's a very simple technique, it's just kind of repetitive, but you do have to make sure you put the right components in the right order, otherwise you'll screw up and have to start over. Once you're finally finished, you're going to let it air dry, or you can blot it dry. Just put 1 drop of water on top of it and put a cover slip on top. Put it underneath the microscope and you should be able to see the bacteria underneath. But you will need to use higher magnifications, and I'll show you guys how to use immersion oil so that you can see things in very high magnification. And I'll demonstrate that in just a second. And for you online people, that'll be in the separate video. So for what I recommend you guys do during that initial step where you're waiting for the water to dry after you get the inoculating loop full of bacteria and rub it onto slide, while you're waiting for them to dry, I recommend you start streaking your McConkey plates with the same bacteria that you are going to be looking at under the microscope. Again, it's McConkey's agar, it's differential media, it's got neutral red, which is a pH indicator. Lactose fermenting bacteria are differentiated. If you guys remember, have you guys taken biochem yet? Biochemistry? Or, well, if you took the basic bio course, you guys learned about respiration and glycolysis and the TCA cycle. So if you remember, the TCA cycle is dependent on oxygen. But if you run out of oxygen, then you have to start fermenting some of the molecules in order to get your ATP, your energy. The thing is, once some of those molecules start to accumulate that you get through the fermentation pathway, is that they're very acidic. So those acidic molecules are going to change the color of the media, and that's how you're able to differentiate between which bacteria ferment and which do not. So the bacteria that are able to ferment are going to turn pink in color because those fermentation products are very acidic and they change that neutral red into red. So as far as the process is involved, though, it's just treat it like you're inoculating or streaking any other plate. It's just the media that's different this time. So I'll go over how to do that again, because I was looking at some of your plates and some of you are still having a little difficulty in getting getting separate colonies when you streak. So I'll show you guys how to do it properly. And then the third thing that you're going to do, so do the streak while you're waiting for your slide to dry off. We're going to be doing step 3 of Koch's postulates. So what is step 3 again? Koch's postulates? Re-inoculate, right. So we got our pure cultures, and now we're going to be re-inoculating a potato slice to see if we can get the same result as what we started off with. That'll be step 4. So let's see. We're going to be doing what's called the stab and swab inoculation technique. Basically all it is you stab the potato with a sterile needle, and then you get some of the bacteria with your sterile inoculating loop, and you rub the bacteria inside the stab wound, just so that you can, that's just the inoculation. It's pretty straightforward, and I'll show you guys how to do it in the technique course. We're also going to be using a negative control. So for most of our experiments, we need to have controls to see, they're in place to ensure that what we see is indeed caused by what we're testing and not by some other reason. So for example, if we start rubbing the bacteria onto the potato slices, and then I take a potato and I also rub the wound that I created, and this time without bacteria. So you got a potato with bacteria and a potato with no bacteria. But then you see both of them start to get that disease, not disease, but they start to rot. What does that tell you about the bacteria? Can we say with certainty, oh, this rotting is caused by the bacteria? No, you can't, because if the bacteria caused the rotting, then why do you see rotting in the potato where there was no bacteria? So that's your negative control. So if all goes well and the bacteria that you guys have is indeed what causes that disease, then you'll only be seeing the spoiling or the rotting of the potato in the potato that gets inoculated with the bacteria. You guys understand? Any questions then? So you guys can do that after you do your gram stain. So we want you guys to work with a partner. Each of you at each desk is going to do just 1 gram stain. Unless you guys want to do 2, I don't mind. You will need to pick only a single colony though from each of your plates. Let's see. Alright, so I mentioned that. For students. So yeah, and if you have different types of colonies on your plate, like if you have 2 separate pure cultures, then you will inoculate 2 different McConkey plates, as well as 2 different potato slices. And that's it. That's all I have for you. Do you guys have any questions about the actual lecture portion? All right. If not, we'll get into the techniques part.